Hello, this is Lauren Feddy, welcoming you to this bite-sized bio webinar, which today is sponsored by Molecular Machines and Industries, or MMI. MMI is a pioneer and global leader in single-cell isolation technologies, providing automated laser microdissection systems for isolating cells, larger regions of interest, and subcellular compartments, while preserving tissue integrity. By combining cell segmentation, high-resolution imaging, and gentle laser microdissection, MMI accelerates discoveries in fields from fundamental cell biology to advanced spatial proteomics. Today's presentation is titled Decoding Early Disease Risk Using Region-Specific Genomics and is being presented by Laurel Hyatt. Laurel is an MD, PhD candidate in human genetics at the University of Utah. Supported by an NCI fellowship, they explore how mutational patterns in normal colon tissue influence colorectal cancer. Their bioinformatics work spans short tandem repeat expansion disorders, pediatric rare disease diagnosis, and health disparities in transgender populations. Laurel serves on our Genetic Society of America subcommittee and anticipates a career as a physician scientist in pediatric medical genetics. As always, we will have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears at the top right of your screen, and I'll put them to Laurel at the end. Details of how to access the on-demand recording of this webinar will be sent to you by email shortly. So now, over to you, Laurel, for the presentation.
Thank you for the introduction. I am Laurel Hyatt from the Quinlan Lab, and today I'm going to be talking about decoding early disease risk using region-specific genomics. I don't have any financial relationships or conflicts of interest to disclose. I'm just here to talk about my research and the ways in which I'm able to do it. I am Laurel. This is my dog Aries. And we have spent the past several years in Salt Lake City as part of the MD PhD program at the University of Utah. I am currently completing my dissertation in human genetics, and I use the MMI cell cut in my research, which has started the relationship and friendship that has allowed me to present at this webinar today, which I'm really excited about. As a future physician scientist in my MD PhD program, I think a lot about disease and especially disease onset. And I find that quite often we think about health and disease as a very binary two-state situation, when in reality it looks more like the bottom figure in which there are several gray areas in the progression of a disease state prior to when we actually think of pathology occurring. And so while there's a lot of clinical focus on the treatment and management of disease, I find myself drawn to this earlier opportunity for screening, surveillance, and intervention prior to the disease onset. And so that can be impacted by mutations, which is my field of interest as a geneticist, which can vary across the population, where different groups have different mutations that make them more likely to experience disease. It can vary across generations, where perhaps a child receives a new mutation that is linked with disease, or it can even vary across an individual, where different mutations that occur in specific parts of the body confer a specific disease risk. And so these are kind of categorized as population genetics, pedigree genetics, and somatic genetics, and I work specifically in the somatic genetics space. And so we refer to this as somatic mosaicism, or the body actually being a mosaic of different genomes. And while this can very often, and actually in most cases, be neutral or not associated with disease, for example, this flower and this dog are both showing examples of actually visible somatic mosaicism. In other cases, it can lead to clinical concerns. The most common case in which we think of somatic mutations is in cancer. It can also manifest in neurodevelopmental diseases, autoimmune conditions, and lastly, we are beginning to understand the ways in which somatic mutations may actually be implicated in the process of aging. It can be, however, really difficult to characterize and investigate and has been challenging until about the last decade or so with technologies. Let's say that the purple here is a mutation of interest. If it is a germline mutation or something found in really every cell in a person's body, we're going to be able to find our mutation of interest in a blood sample, in a skin sample, and really anywhere we're able to collect tissue. Whereas a somatic mutation, if it's here, restricted to the stomach, we would only get it if we actually biopsied that organ, which is obviously going to be challenging depending on what your tissue of interest is. Furthermore, it can even vary within tissues. And for example, recent studies have shown that actually where a mutation arises within the brain and how it spreads within the brain can impact the resulting pathology. And so you can have half of the brain being affected in a dispersed mutation with a condition known as hemimegalencephaly, or it can be restricted to just a lobe or even a small portion of the brain, which of course makes sampling those tissues really important in considering the specificity of what you're actually getting to examine. And so one of the questions that really inspired my thesis work is if these regional mutations lead to regional manifestations of disease, could the inverse also be observed? If we look at cases of regional disease, would we then see also regional mutations and mutational landscapes? And so the context in which I think about this is colorectal cancer specifically. And so it's known that different mutations really, really characterize the different kind of archetypes of colorectal cancer that we've observed clinically. And these impact the prognosis of the individual, what treatments we consider, as well as how closely we will likely follow that person clinically in the future. And one of the things that I found really fascinating in my medical school years is that colorectal cancer both has distinct regional clinical manifestations, but also the somatic mutations themselves we found to be region specific. And those in turn associate with the clinical characteristics I've just discussed. And so the real motivating question for my thesis work is what we can learn by interrogating regional somatic mosaicism in specifically pre-malignant colon or colon before the cancer has really um, occurred. And so, again, thinking about this pre-disease stage where we have the opportunity for clinical intervention and combining that with the regional resolution, which we know to be clinically salient. And so, with these combined, I'm really interested in looking at the region-specific histories and trajectories that mutations can inform us of and where those might be clinically relevant, which is especially pertinent because actually early onset colorectal cancer has been rising in incidence and mortality despite colorectal cancer overall having improved clinical outcomes. And this has been found to be specific to left-sided or especially rectal colorectal cancer. And since we don't currently understand the mechanisms leading to this, we're not in a position to really address this public health problem. And so, as you'll recall, somatic mosaicism can be challenging to characterize based on our access to tissue. However, we at the University of Utah are in a unique position to actually assay regional colon. And so the University of Utah, where I study, does more than 10,000 colonoscopies per year and actually has started a biobank called the Magic Biobank with director Dr. Amiko Uchida to actually combine endoscopic procedures with the storage of tissue for research purposes, which has been a phenomenal partnership that I'm very lucky to have been able to use as part of my PhD. And so from there, we have worked with gastroenterologists to actually take 5 to 6 regional biopsies based on anatomical landmarks. And if you love colon anatomy, Those are through the cecum, ascending colon, transverse colon, descending colon, sigmoid colon, and rectum. This will not be a test question. You don't need to worry about these, but just know that we're taking really standardized biopsies across the colon at these regions consistently across different patients. And again, in the context of these pre-malignant or pre-disease cases, we consider healthy individuals to be individuals with no findings on colonoscopy compared to individuals where we find polyps, which we know are conferring some amount of potential disease risk. And so we've endeavored to have 10 patients for each cohort. We ended up with 10 and 11 because we're so lucky, and moved forward with this really wonderful cohort of tissue samples. Now, you'll recall that somatic mosaicism also varies within tissues, which can be a bit of a headache. And so, despite having these bulk biopsies of these specific regions, we have to ask ourselves, okay, well, how do we make sure that we get the relevant cell type? And so, for us, that is the colon crypt, which is the functional and experimental unit of the colon. Each one is about 2,000 cells and kind of looks like a test tube from the side. There's about 15 million per colon, so quite a few. And I believe the factoid I found out recently is that we create more colon crypt cells in our lifetime than there are like galaxies in the universe, which is pretty incredible. There's a lot of these cells. Um, and when these become dysregulated and grow without control, this is actually, we've discovered what leads to colorectal cancer in the conventional form that we think of it as. And so they're very clinically relevant as well. And the way that they're structured is they have a little stem cell niche at the bottom that self-renews and then proliferates. It travels up the test tube, differentiates into the absorptive cells, and then sloughs off, disappears off into the digestive tract at a scale of about every 3 days, which is very quick for the body. And so most stem cell mutations or things that happen in the stem cell and then carry through the differentiating cells are known as clonal mutations, or present in about half of the DNA in the crypt. And so this is really exciting because very often we use things called clonal expansions, where we grow out cells that are similar, which makes it easier to evaluate these mutations. And the colon crypts actually form these naturally without us needing to grow the cells on our own. And so they're a really exciting experimental model if we can just get to them. And so the way that I actually do that is with the MMI cell cut. And so I have a video here at 2 times speed, which I got quick enough to do this in real time. And you can see I basically trace the crypt and then use the laser to actually cut around it, which becomes a little bit more clear as we cut through more of the colon crypt. And then once we have outlined it fully with the actual laser, it is removed into the capture tube where I can then use it in downstream experiments. And hopefully it's okay to just mention that I'm very, very grateful for this technology. The technical aspects have been really instrumental for my project. The laser, after demoing several systems, was the one that actually maintained the best DNA quality, which is essential to the work that I do. And I've also just really appreciated working with the folks at MMI. And so just taking a brief second, if it's allowed to just say that I'm very glad to actually have a technology that has facilitated my thesis work. And so it is one part of this broader workflow that I have been using, which involves also low input library prep and element whole genome sequencing. I'm happy to take questions about these aspects, but we'll sort of summarize by saying that my goal was to combine these different technologies to really optimize our workflow to capture crypt mutations at the highest quality. And the reason for that I can explain by sort of turning the crypt on its side. And so again, we have these stem cells at the bottom and the differentiated rings of cells towards the top. And we have the cell lineage, right? The history of the cell, what led to that cell as it populates through the broader ring of the crypt. And when we have mutations from the stem cells, they're generally found in all of the cells traditionally, but the actual practice of this, and I'll again color these in one color just for simplicity, um, helps us confirm these mutations in what are called reads, or basically the parts of the DNA that we're looking at when we're sequencing it. And this gives us a theoretical variable fraction of zero point 0.5, or again, in half of the DNA. And if you see something in half of your data, you're able to pretty confidently say, okay, that thing probably exists, which is the benefit of looking at these clonal mutations and why they are so prevalent in current studies of colon crypts. However, necessarily, not every mutation is going to be subclonal or derived from the stem cell. You're going to have cases where in this niche of anywhere from 4 to 12 stem cells, only some of them have a specific mutation, which then might differentiate into that top ring of absorptive cells. Or you may have a mutation that occurs during the actual differentiating process. And so it isn't from a stem cell, but still persists at this top kind of level of the colon crypt. So this was something for my project where when I'm trying to look at these different really trajectories of mutations in colon crypts, I was really interested in optimizing the capture of subclonal mutations, since, more data gives us more opportunity to interpret the histories of these colon crypts and to see the types of mutations which might more conventionally be repaired in stem cells. And so that was part of my project in trying to find the gentlest laser, the you know, most robust library prep opportunities. And so the question that we've had is, how well are we doing capturing these subclonal mutations? But first, I'll address how we did overall with our experiment. And so as I mentioned, we have two cohorts that ended up with 10 and 11 individuals, so 21 donors overall with about 125 crypts. And I've been benchmarking my data set against a published data set from 2019 looking at colon crypts from 42 donors, not with the same regional resolution, but a similar experimental design of a individual colon crypt that was laser captured and then whole genome sequenced. And so you'll notice I have the red of my name Hyatt for our data, since we are at the University of Utah, which is red. And then I'm comparing to data set from the Sanger Institute, which has a blue color scheme. And so that will be the recurring theme. And so comparing coverage at the threshold of at least 10 reads per base pair or a specific region in the genome. Again, my data is going to be in the red. All of Elise data is going to be in the blue. You can see that our average coverage is about 10x higher than the Elise data set. And we were able to even get some above 50x in our experiments. And again, the more coverage we have, not only the more confident we can be in our mutations, but the greater likelihood we have of capturing these subclonal mutations with confidence. And so I was pretty happy with our success on this front. Additionally, we look at the actual base quality, right? How much do we believe the mutations at any given base in the genome? And so we're using these two metrics, Q20 and Q30, which correspond to 99% accuracy and 99.9% accuracy, respectively. And so when we look at our data as well, by these two metrics, what we see is with us in the red and the Lee6 data in the blue, we have a very consistent, high quality base quality, which is great because it means that we are more able to trust the mutations that we are calling in our data. And so again, was pretty excited about this. And so when I am looking at the actual amount of subclonal mutations that we were able to acquire in our data set, uh, variant allele frequency, again, is just showing the percentage of all of the reads that we were looking at, how many of them have the mutation, and so, kind of in the middle here is where we consider our clonal mutations, and then subclonal is going to be a smaller fraction, so off to the side of that. And so, comparing again my data in red and the Lasix data in blue, you'll see we have a lot of overlap um, around to where we get to the clonal mutations, which makes sense because of the known biology of these colon crypts. But then when we move over to the subclonal side of things, we see a lot more of these red subclonal mutations, which has been really exciting for my data set to be able to look through, again, these non-stem cell associated mutations in the data. And so there's a lot to unpack, you could say, with this new data set, which has been a lot of fun and a really great opportunity for me and is only really made possible by, I think, the technical aspects of my workflow, which again, I am very grateful for and was able to optimize with the help of a lot of wonderful folks such as MMI. So um, and then the question, of course, is how does the biology look if we're capturing more subclonal mutations? And the good news is that our mutation types are still consistent with Lee6 and the known biology of the colon crypt. And so if we're looking at these mutation types, and so this is where a cytosine, for example, mutates to an adenosine, we can collapse basically all of the single one base pair mutations into these 6 mutation types, which are often used to compare overall mutational trends in different tissues. We can then compare the proportion of these, right, to see how common one is versus another. And what we end up seeing in the healthy males across our data and the Lee6 data set, that the cosine similarity, which basically just suggests how similar these two spectra are, is about 0.981, or you can also interpret that as 98% similar. And so while there's some amount of increased variation in our data set, a lot of it driven by those subclonal mutations, the overall biology tracks with what we would expect from these colon crypts, which is great, which is, of course, a relief. And so, um, again, returning to my kind of thesis question of what we can learn by combining the pre-malignant colon with this regional resolution, we now have data that we seem to be able to trust, and that follows with what we understand about colon crypts. And then in order to get at this question of these region-specific histories and trajectories, we use something known as mutational signatures in the field. And so a mutational signature is just a way of inferring mutation etiology, or to put it a little bit more simply, we use what we see now to infer what has happened before. And so the example I'll give is you may remember Aries, my lovely dog, from the very beginning of my PowerPoint. And she is a lovely little angel who is also a little bit rambunctious. And so if I were to come home and see her like this, I might look at what is currently measurable and say, all right, it looks like there's some peanut butter on her face, and so she was able to get a little peanut butter treat. I see the red on my toes, which suggests that maybe she walked in red paint. And then I see a little bit of grass stains on her back, so it looks like she had some fun rolling around in the backyard. And so, with that similar line of reasoning, we can look at again these single base substitution signatures or the single nucleotide mutations, like I mentioned before with the spectra, um, C to A, C to G, and so on. And again, look at where they are. And so we look at them in the context of the single nucleotides on either side. And so for the C to A's, we can look at them in the context of where there's two adenosines on either side of the C, and there's about, or there are exactly 96 configurations of these in total. And again, we can look at the different proportions of all of these specific mutation types. And here we have single base substitution 18, or the signature 18, which we know to be associated with oxidative damage. And so basically, we look at these different mutation types and where they are and what frequency they occur at, and we can correspond those to what we know about different mutational uh, processes in order to kind of build an understanding of what has happened with different tissues. And so they are a massive deal in colorectal cancer research. I've just included one review here from a couple years ago, but mutational signatures have really been leveraged in a lot of somatic mosaicism research, and especially in, again, colorectal cancer studies, as well as specifically these E. coli related signatures have been very utilized in colorectal cancer research. And so specifically, there is a type of E. coli called PKS plus E. coli, which creates a molecule known as colobactin, um, which looks like this. It was discovered somewhat recently. And that colobactin loves parts of the genome that have a lot of A's and T's. And so it basically will form an adduct where the molecule basically binds loosely but preferentially to those regions in the genome, and then will cause mutations that have now been associated with two signatures. This single base substitution signature 88, as well as a insertion deletion signature ID18. And so again, just drawing your attention to where it has these A to T rich segments, we look over and it's really these T to C mutations we see in SBS 88, and then these T deletions that we see in ID18 that we've been able to correspond very closely to this particular type of E. coli. And there is a lot of emphasis that has been placed on colobactin in recent colorectal cancer studies. It has been suggested to be a cause of adenomatosis polyposis or colon polyps in colorectal cancer. It has been linked specifically to the early onset colorectal cancer phenomenon that I mentioned earlier. In this opinion piece, they actually describe the identification of colobactin as a major step forward, addressing the public health challenge. And then another recent paper has suggested that it's actually one of the major causes of colorectal cancer. And so I really want to emphasize that this has been a major aspect of the hypotheses around colorectal cancer development. And so, of course, my question as a future physician scientist is, can we use this aspect from research in the clinic? And so could we potentially use it as a prognostic biomarker? And especially there's a lot of data suggesting that it happens in early life and that it might predispose people to colorectal cancer later on. And so the question becomes, of course, is this something that we can use in the context of screening and early intervention for individuals if it really does confer this type of disease risk? And so I compared our data set of individuals with no findings and polyps to an actual published data set of colorectal cancer tumors. So specifically, the Cornish et al. 2024 paper uses 2023 tumors, which have regional annotation, which is great for our regional comparison. And if you recall these regions, I'm now going to flatten it on one axis so that it looks like this for these coming figures. And so to orient you, first I'm going to show you the signature SBS1, which is involved in the spontaneous deamination of cytosine to thymine or C to T mutations. And so, again, comparing these three cohorts, healthy at risk and the Cornish tumor cohort, to our scale, because generally a non-tumor sample is going to have far fewer mutations than a tumor sample. And so we have two scales. And then we are looking again at these different regions. And so here, what you can see is the trends are shown by the different colored lines. And then I have the standard deviation as these error bars. And the SBS1 signature is concordant with the cancer and the at-risk cohorts, where it seems to be highest in the cecum or at one end of the colon, and then generally decreases, which is not the case in our healthy, slightly younger cohort. And so I'll draw your attention that we're comparing on one side 0 to 80 mutations and on the other side 0 to 4,000. And so our scales are quite different here, but we're able to evaluate the overall regional trends, which again gives us information about what has happened and what might happen in the future for these individuals. And so now I'm going to show you the colobactin signature ID18 by clinical cohort. And you can see that these lines are actually very, very close together. Um, but what I want to draw your attention to that was pretty startling for me is that the scales for these plots is actually the same. Not only are the trends concordant across the three clinical cohorts, but actually the mutation burden itself is pretty identical by region, which kind of blew my mind to see when we first looked at this data set. And we're towards the end of the presentation, so I'll just summarize some of the work I've done to really evaluate this. And so I looked at whether the Lee-6 data set matched the burden as well. It does. I looked at whether a secondary tumor cohort that came out also fairly recently matched this. It does. And then I looked at the sequences rather than the signature inferences from the Huber et al. Paper that came out a few years ago, where they looked at colobactin affected organoids and then the sequences, the AT-rich sequences affected, and found that those also match regardless of clinical status, which was fairly surprising. But all of the data seemed to converge that these mutations seem to happen early in life and consistent, really regardless of clinical status or outcome. And so our conclusion is that these are possibly misleading biomarkers. It's possible that this is a wrong place, wrong time situation where these colobactin mutations preferentially occur in the rectum because that is where this strain of E. coli um, has sort of the best of both worlds with regard to the oxygen levels and so forth in the colon. And because that is also where early onset colorectal cancer is happening most often in the rectum, there's been a kind of linking of these phenomenon that may not be as causative as we previously thought. This follows with actually a study published just a few months ago that showed more than half of infants followed for 24 months actually had some amount of this E. coli strain colonizing their intestines, where they used the phrasing further investigation is required to understand the circumstances in which carriage can lead to mutagenesis. And I think this really gets at what I found to be the core of my experience in my dissertation work, which is we really need to understand our mutational baselines before we can assess disease risk. And I think that as a field, we have spent a lot of time studying disease tissue, and we have a real opportunity now with our current opportunities of technical and computational advances to really look at pre-malignant or non-disease tissue to understand, where we start to understand how we get to disease. And with that, I've got to say thanks to everyone involved. I have a wonderfully supportive lab and a wonderfully supportive research environment. I'll also shout out Xiaoshu Yang in particular, worked with me to get the research grant to bring our cell cut to the University of Utah, which again has been instrumental for my thesis. And so I'm also grateful for your time and attention, and I am happy to take any questions at this time.
Thanks, Laurel. That was a really great presentation. And we have a few questions from the audience. If anyone else does have a question, please feel free to post it in the questions box at the top right of your screen. Okay, so the first question that we have, what are other tissue or disease processes where regional analysis could make sense?
Hmm. I think, I mean, the easy answer is everything, right? But I think any tissue system in which there are going to be functional differences and therefore different um, consistent exposure leading to different mutagenic processes, I think would be particularly interesting. And so even just staying in the cancer space, there's differences right in like parts of the stomach. We're still even in the GI tract, right? And so I think that evaluating really based on like the functional aspects of organs is one way to start. But then when you have different techniques, right, that can process low input with different cell specific types, I think there's just like a whole world of opportunity. And so I think the colon is helpful because of the colonoscopy and like the accessibility of tissue. But I think with, increases in like biobank infrastructure at a
Yeah.
lot of institutions, I think there's just tons of opportunities, which is exciting.
Okay, thank you so much. Um, would it make sense to approach this study from a non-colon crypt lens, like single cell or organoids?
Mm. Um, yeah, I think that it makes sense depending on what um, questions are being asked. And so there's been previous organoid studies growing from individual like adult stem cells. And so I think that it's kind of the two ends of the spectrum of if you're interested in these clonal and potentially early life exposures, then an organoid could make a lot of sense interrogating those mutational dynamics.
Yeah.
Whereas single cell experiments, I think, can lead themselves more to these subclonal or really like sporadic mutagenic etiologies. But I think a lot of the field has gravitated towards colon crypts, and that's ultimately what we chose because it offers right
Yeah.
that middle ground of a monoclonal structure where you can also potentially get some amount of subclonal information.
Great, thank you very much. What about considering epigenetics, methylation, or other related mechanisms in this context?
Mm. There's some amount, I think, of like rumbling in the literature about especially like RNAs in the context of colorectal cancer
Yeah.
biomarkers. And so I think that it would only, add more information. I think some of the, technical challenges involve the DNA input to, what is required for getting, multi-omic information. But again, that's somewhere where potentially like a clonal organoid could be leveraged, um, or, trade-offs can be made to pursue it. I
Yeah.
think it would be clinically and, biologically informative. But I think some of the, again, experimental trade-offs are why we chose the route that we did.
Yeah. That's great. And have you identified any mutations that provide beneficial effects rather than causing disease?
Mm-hmm. Um, I really love this question because it asks the question of, what do we consider beneficial, right? And so something that comes up a lot in somatic mosaicism, or really I should say maybe some mosaic research, is things that are beneficial at a local level, but not necessarily for the organism. And so I think
Yeah.
of my lab has studied selfish mutations in sperm, which are great for, stem cell proliferation, but confer disease on the offspring. You
Mhm.
contrast that with there's been mutations identified in liver that are actually beneficial to the entire organism because they lead to regeneration, which
Mhm.
is, helpful in context of disease. So I, in our project scope, we've really focused on mutations that are potentially disease conferring at the level of the
Yeah.
organism. And so, in that context, we haven't identified really any protective mutations. But I do think it's something to consider in the balance of we've identified potentially mutations that are um, like pro-proliferative at the level of the crypt, which is,
Mhm.
great for the crypt, but less so as the individual when that potential growth can lead to the
Yeah.
dysregulation that we know to be pathogenic. So, not in our study, but I think even asking the question forces us to think about like how we're really framing the consequences of these mutations.
Yeah. Okay, that's great. Thank you so much. So that brings us to the end of the webinar. Thanks again, Laurel, for a great presentation and great discussion.
Thanks for
Thanks
having me.
also to our sponsor, Molecular Machines and Industries. And finally, thanks to you, our audience, for taking the time to attend and listen in. So until next time, good luck in your research and goodbye from all of us at Molecular Machines and Industries and Bite Size Bio.
Thanks so much. It was great to be here.